Anna Maria is from the MD Anderson Cancer Center, and going to talk about this project to develop an immunology ontology. Thank you.
Yes, and so really today is the introduction of this effort that we are starting to develop an immunology ontology. As you said, you see in this title is trying to fill in the gap. So most of you would say, you know, why an immunology ontology? We already have ontology, particularly in OBO ontology that covers immunology. You know, we have CL, gene ontology, OBI, VOR. Some of them they already cover a big part of immunology. But what they cover, I would say, is the main general immunological information. What is still missing is the context. And in immunology, the context is really a main part of information, because it's not anything that happens with our immune system is not static. First of all, the cells are moving, they are doing things all over the body, and they are doing different things depending on which is the context. And so that's what we think is missing to really well represent the immunological data, is really given context. So telling where, when, and what is happening to get that results. And so that's what the immunology ontology that we started, we want to develop, is going to aim to do. Really create a possibility to have a context model to represent the context for each particular case of the immunological response. And as I said, oops, sorry. Why is going back? Sorry, I don't know why. Let me go back. It's going. Okay. And these are just a few examples that I want to give you here, is just to show you how really the context makes the difference. So here is, you know, the same cell producing the same molecule like interferon type 1 can have completely different results depending in which context that cell is producing that molecules. And you can have really the protective effect when you're talking about something where you have an infection, as well as you can have a continuing activation of immune cell and developing autoimmunity, or on the other hand, having, depending on the timing and the concentration, you can have, a response to cancer or a completely tolerance to cancer. So really the context makes the difference. And so we need to capture that part in order to understand what's going on, in particular, in order to be sure that when we are reasoning over data, we can get really the very specific outcome. And these are, today my computer is, I don't know what's wrong. And these are just other few examples, like this is another molecule, says the chemokines, and you can see that I don't want to go through all this, obviously, but you can see that you can have a niche retention role. So it is not going to move cells if it's happening in the bone marrow, as well as instead is going to facilitate metastasis if it's going to happen in the tumor. So it's going to create, even in some other case, the barrier for lymphocyte to get in. And so the effects are different depending where and when and in which context they are happening. And the same for this is the receptor, that chemokine, that depending in which is, again, the context, is going to have one effect and the opposite effect too. And so if we are now going to capture this context, then it's going to be very difficult to really be able to understand, because for the same thing we could have, as I said, annotation that says it does something, but it does even the contrary of that something. And so how we weigh that part. And so we really need to cover the context. What we are thinking to approach it is not obviously not taking account what has been done. It has been done a huge amount of work on all these other ontology that cover that part. But as I say, those representation are still separated because each of these ontology that cover that part of immunology really do not have the immunology as their main domain, but they use and they represent immunology in other contexts. So, for example, for representing gene ontology, to representing the gene function of processes as well in the cell, which part they have in immunology, but it's not the immunology itself the main domain of coverage. And so our idea is that to create this immuno that is going to work as an umbrella where we will bring in all the terms that has been already developed in other ontology so that they will be under, be able to be searched under one ontology, and at the same time create the specific relation that will represent the specific context inside this ontology. So that is really what we don't want to replace anything. We really want to reuse anything that is there, but just create a unique umbrella under which all these things will be used to create a context. And we have started. I want to say that the idea is not to only cover disease, but even cover physiological immunology. But we have started because we have the opportunity to start with the autoimmunity. And why autoimmunity we consider is a good start, because really has a rich context availability. Is not there is not a clear and well-defined representation of what's going on during the autoimmunity. We will obviously reuse a lot of this ontology that cover part of this immunological. Then we have data because we rely on the autoimmune registry cohorts. And in particular, we decided to start as a use case with the Sjögren disease. And the idea is to start from this autoimmunity and to create a model for context, to represent context that will be used not only obviously for the autoimmunity, but for all the other immunological contexts. So really the idea is to use this use case of Sjögren to develop a context model for immunology. And obviously that will be used, as I said, for other cases, like we have already idea to go to the cancer immunology, as well as the physiological neonatal immune development, and as well as all others. So really should be one model that could fit all the possible use cases. Obviously, one of the things that we need to account are obviously which are the advantages and the risk. The advantages to have this kind of representation will be that we will have obviously fewer ambiguous labels, and in particular the ability to go cross-study cohort retrieval, because we are, be able to represent the context and have a clear model for that. The risk on the other end are that since we want to cover a large and complex domain, we could kind of lose the focus and kind of be diluted in all these things. So that could be one of the risks. The other risk is that we could overlap with some ontology. And the other risk is that could be the sustainability, because it's going to be a large kind of representation in order to do. So that could kind of be part of the risk that we need to take in account when doing this. But the reason, so I want to, first of all, this is our work. So it's obviously it's not me, it's all these people that has started this. And as I say, this is just the beginning, and we just started to talk, and we just to define our use case. But we need, in order to do it right, really we need to have more people involved in this. And that's why I hope that there will be other people that would like to join us to develop this immunology ontology. And we really need, people from different part of the community, so immunologists, pathologists, ontology, data steward. So all of this different view of the same problem will really help us to create that model, context model, that could really be able to fit all the immunological context. And so that's, I hope that here is, there is people that would like to join us. And with this, I really thank you for listening.